Good evening, good morning, good afternoon to the audience that watch this video. This occasion, I would like to present about the task of biochemistry number 4 about metabolism second. First of all, my name is Wayan Ananda Savitri with last name number 7. And let's move to next slide. The question number 1. First question. Explain the main function of the citric acid cycle, Krebs in energy metabolism and its relationship with acetyl coenzyme A. The citric acid cycle or Krebs cycle takes place within the mitochondrial matrix of the cell. We can observe that highly active cells such as muscle or nerve cells contain a very high density of mitochondria. The primary products of this cycle are not large amount of ATP directly, but rather high energy electron carriers, namely NADH and FADH2. Furthermore, the cycle requires acetyl coenzyme A as the primary fuel to initiate the series of reactions that drive the cycle. And then, why is acetyl coenzyme I so crucial? Acetyl coenzyme A, a two carbon molecule, acts as the bridge connecting the metabolism of carbohydrates, fats, and proteins to the Krebs cycle. The cycle cannot proceed without acetyl coenzyme A, which combines with oxaloacetate and four carbon molecule to form citrate, a six carbon molecule. The primary function of this cycle is not to directly burn glucose, but to extract as many electrons as possible from the acetyl group to be transferred to the electron transport chain. The relationship between the acetyl coenzyme A and the Krebs cycle can be modeled as a simple flowchart. Acetyl coenzyme A plus oxaloacetate it makes or produces citrate. The process the cycle a series of oxidation and decarboxylation reactions. And primary output is first high energy electrons, 3 NADH and 1 FADH2. Second, directly energy, 1 GTP or ATP. And the last byproduct, 2 carbon dioxide released as respiratory waste. The primary function of the Krebs cycle is to act as a central electron collector. Without this cycle, the energy stored in acetyl coenzyme A could not be efficiently converted into ATP. Acetyl coenzyme A serves as the entry point, ensuring that carbon atoms from various food sources are eventually broken down into CO2 or carbon dioxide, while the released high energy electrons are captured by NAD plus and FAD to form NADH and FADH2. These electrons are subsequently used in the inner mitochondrial membrane to create a proton gradient, which then drives large-scale ATP synthesis through the process of oxidative phosphorylation. Question number 2. Describe the main products of one turn of the Krebs cycle, ATP or GTP, and ADH, FADH2, and explain their role in cellular energy production. In one turn of the Krebs cycle, which possesses a single acetyl group, we can observe systematic chemical changes. Through laboratory observation of mitochondrial activity, the release of carbon dioxide CO2 and the formation of energy carrying molecules are detected. Specifically, observation of the energy produced is not entirely in the form of ATP immediately. Instead, the majority is stored in the form of reducing molecules. Why does this cycle produce more NADH than ATP? This is due to the cell's efficiency strategy. While one ATP molecule provides instantaneous energy, NADH and FADH2 act as rechargeable batteries that carry high energy electrons to the next stage. Oxidative phosphorylation, by reasoning through this mechanism, we understand that without NADH and FADH2, the cell would not be able to produce the large amount of ATP required to support the feeding function of complex organisms. Alright, this is the table of the uh, product and the primary role. The first one, NADH, the amount of molecule is three molecules, and the primary role is the main electron carrier to the electron transport chain. And then FADH2, it contains one molecule that the primary role is supporting electron carrier to the electron transport chain. ATP or GTP contains one molecule, and the primary role is chemical energy ready for cellular work. And then last CO2, it contains two molecules, and the primary role is carbon byproduct during respiration. In a sense, the Krebs cycle serves as the primary supplier for oxidative phosphorylation, while the ATP or GTP produced provides immediate energy. The NADH and FADH2 function as vital electron carriers that drive the electron transport chain, triggering the ATP synthesized enzyme to generate the bulk of cellular energy through oxidative phosphorylation. Question number three. How can disruption to the Krebs cycle lead to reduction in energy ATP production? Clinically or experimentally, disruption in the Krebs cycle, for instance, due to enzyme deficiencies like fumarase or inhibition by toxins, are characterized by symptoms such as chronic fatigue and muscle intolerance. Observations show that when one of the enzymes in the cycle is inhibited, intermediate substrate accumulate, while the production of final products, namely NADH and FADH2, drops drastically. Consequently, cells inhibit very low ATP levels even when oxygen and glucose supplies are available. Why is an inhibition in Krebs cycle fatal for energy production? The Krebs cycle is the primary bridge that converts chemical energy from food sources into electron energy ready to be transported to the electron transport chain. If the cycle is inhibited, the electron transport chain lacks full electron donors without a consistent flow of electrons from NADH and FADH2. No proton gradient is formed in the inner mitochondrial membrane. Without that gradient, the ATP synthase enzyme lacks the driving force to phosphorylate ADP into ATP. Disruption in the Krebs cycle causes a drastic decline in energy production because the cycle serves as the primary supplier of high energy electrons for the electron transport chain without sufficient NADH and FADH2 as electron donors. The mitochondrial proton gradient cannot be established, leaving the cell's main energy producing machinery ATP synthase without this driving force, causing ATP production via oxidative phosphorylation to halt entirely. Question number four. Explain the mechanism by which the electron transfer chain functions in the inner mitochondrial membrane. Alright, this is the mechanism. Uh, First, we got NADH here. In this situation or in this context, NADH release its H plus and then into complex number one, and then it's through this red red arrow to this, and then complex 2, complex by through complex 1, and then complex 2, complex 3, and then complex 4, and yeah, and then it's release H plus and it becomes NAD plus. Why the electron calls the proton? Because when the electrons transfer through a protein complex is exothermic reaction. The energy they release in every step, and to pump proton to against the gradient, concentration gradient from low to high. In this process, it's like charging the membrane. It's just like charging the battery. And then next question, the role. Yeah, an analysis of the roles of NADH and FADH2 in the electron transfer chain and how they contribute to ATP production. The analysis of the roles of NADH and FADH2 reveals that both function as high-energy electrons carrier that donate their electrons into the electron transport chain within the inner mitochondrial membrane. Logical reasoning shows that NADH initiates transfer from complex 1, thereby triggering proton pumping through 3 protein complexes, while FADH2 enters via complex 2, pumping proton only through 2 complexes. This model by an ATP production efficiency of approximately 2.5 ATP for NADH and 1.4 Point 5, sorry, ATP for FADH2 due to the difference in the resulting proton gradient, explaining that both contribute by providing the electron fuel that creates the electrochemical gradient needed to drive the ATP synthesized enzyme in the synthesis of ATP as the primary form of cellular energy. Number 6, explain the concept of oxidative phosphorylation and how the proton gradient is used to produce ATP. Biologically, we can observe oxidative phosphorylation as the final stage of respiration, where energy from electrons flowing through the electron transport chain is used to pump protons H plus into the mitochondrial intermembrane space, such that logical reasoning will demonstrate that this proton accumulated creates an electrochemical gradient, the proton motive force, that stores high potential energy. This mechanism is modeled like charging a battery and is explained as the process of chemiosmosis, where protons flow back into the matrix through the ATP synthase enzyme channel, rotating the protein structure to catalyze the attachment of inorganic phosphate to ADP to form ATP, representing the peak of cellular energy production efficiency. Question number 7. Calculate in principle the amount of ATP produced from one NADH and one FADH2 and explain the difference in the energy yields. Biologically, we can observe that each molecule of NADH generates approximately 2.5 ATP, whereas FADH2 only generates approximately 1.5 ATP during oxidative phosphorylation, where logical reasoning reveals that this difference is due to the electron entry point in the electron transport chain. NADH donates its electrons at complex 1, thereby pumping protons through 3 proteins complexes, while FADH2 enters a complex 2 and skips the proton pump at complex 1. This process is modeled as a difference in proton gradient formation efficiency, proton motive force, which explains that NADH produces a stronger gradient than FADH2, allowing the ATP synthase enzyme to produce more ATP for every oxidized NADH unit compared to FADH2. Next, now question number 8. Explain the process of glycogen breakdown and how this process helps maintain blood glucose levels. Clinically and experimentally, we can observe that individuals with mitochondrial dysfunction frequently exhibit symptoms such as chronic fatigue, drastic decline in stamina, and exercise intolerance. Even during low-intensity activities at the cellular level, microscopic observation reveal defects in the structure of the mitochondrial cristae. While biochemical blood analysis often shows elevated lactic acid level as a compensatory response to the lack of aerobic energy production. Why does damaged mitochondrial structure lead to muscle weakness? Because mitochondria are the cellular powerhouse. If biochemical pathways like the Krebs cycle and the electron transport chain are disrupted, the efficiency of converting nutrients into ATP drops significantly. Muscle tissue is highly energy demanding. The concentration of actin and myosin filaments requires vast amounts of ATP. When mitochondria cannot produce enough ATP to meet metabolic demands, muscle cells cannot contract optimally, resulting in fatigue and functional weakness. Mitochondrial dysfunction in muscle cells can be modeled as a cellular battery failure where structural damage or enzymatic deficiencies directly inhibit primary metabolic pathways, specifically the Krebs cycle and the electron transport chain, leading to a drastic decrease in the production of ATP or cellular fuel. The inability of mitochondria to provide sufficient ATP for the concentration of actin and myosin filaments creates a chronic energy deficit with the muscle cells, which ultimately manifests microscopically as mechanical muscle weakness and persistent fatigue. Biochemically, mitochondrial disorder causes a failure to generate the proton gradient necessary for ATP synthesis through oxidation phosphorylation. Consequently, muscle cells are forced to switch to anaerobic metabolism, which is far less efficient and produces lactic acid. Because the muscle cells cannot maintain sufficient ATP levels to sustain the mechanical strength of actin and myosin filaments. The physiological manifestation is significant exhaustion and chronic muscle weakness. Question number 9. Explain the factors that regulate the rate of the Krebs cycle and the electron transport chain, e.g., the ATP or ADP ratio and ADH or NAD plus ratio. The rate of the Krebs cycle in the electron transport chain is tightly regulated by substrate availability and cellular energy demands through negative feedback mechanism, where a high ATP or ADP ratio indicating an energy-rich state inhibits key enzymes such as isocitrate dehydrogenase in the Krebs cycle and slows down electron transport chain activity to prevent energy wastage, while a high NADH or NAD plus ratio signifies the saturation of the electron donor, which also slows substrate oxidation. Conversely, when ADP and NAD plus level increase due to high energy demand ATP consumption, this inhibition is released, thereby increasing the rate of oxidative metabolism to accelerate oxidative phosphorylation and mid-cellular ATP requirement. The last question explains its relationship between glycolysis, Krebs cycle, electron transport chain in the overall production of energy within the cell. Alright, first we go through glycolysis. Glycolysis happens or occur in cytosol. The the first phase or the beginning phase, it break down to one molecule glucose or six carbon into two pyruvate. Yeah, pyruvate glucose break the glucose into pyruvate, and then produce a little bit of ATP. And then the specific role or the primary role is to develop unit carbon that's smaller and produce some NADH that has function to 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 carry the electron. And then Krebs cycle, after pyruvate got into the mitochondria, this cycle acts like carbon production and systematically takes the high energy electron from the carbon molecule, and the product is CO2, but the main product is NADH and FADH2. Oh, this is not contained FADH2. And then it carries a high energy electron And then transport electron chains and the ATP, the ATP synthesize NADH and FADH2 that produce and then give the the electron into the transport chain. And then ATP synthase enzyme use the energy from this proton to change ADP into ATP in high or big amount. All right, that's all. I would like to say thank you to the audience that watched me until the end. I would like to say thank you, and I would close this presentation. Thank you.